Welcome and good morning. And so in mass spectrometry imaging, increasingly we are being asked to identify where within a cell a compound is located. And so historically we've been able to look at single cells with TOF-SIMS imaging, and we can see compounds inside the cell, but people want to know, okay, what specific organelles are these compounds located? And in particular, people are looking for drugs, drug metabolites, things like that. And so up until now, there has not been a good way to know for sure which subcellular compartment specific compounds are located in. And Mary Kraft, a collaborator, had a quite elegant idea to achieve this task, to define subcellular organelles and put a tag or a marker so that we can identify where we're looking at within the cell. And so I'll get to that in a few slides. Starting off, I'd like to acknowledge my collaborators. So the group of Professor Mary Kraft at Urbana-Champaign and her PhD student Corinne Chinney. And at Colorado State University, the group of Michael Tamkin and his PhD student Ben Johnson. So a few years ago, Mary was doing some studies using dynamic SIMS. So this is elemental capability, imaging mass spectrometry. And so she was looking at a problem or addressing a question of whether cholesterol is involved in the formation of lipid rafts on a plasma membrane. And so to do this study, she was using nitrogen-15 labeled sphingolipids and oxygen-18 labeled cholesterol, and looking at the distributions of these stable isotopes in these lipid membranes. And so some of that work is shown here for MDCK cells. On the left is the nitrogen-15 enrichment plot from the sphingolipids, and on the right is the O18 distribution from the labeled cholesterol. And so simply put, her conclusion was that cholesterol does not appear to be involved in these lipid raft formations. But she took this analysis a step further, and she looked in depth, so going beyond the plasma membrane. And so the area of depth profile analysis, or 3D information, is shown within the white boxes here. So on the next slide, we're looking at 3 different depths into this MDCK cell. On the top row, we have the secondary electron image. In the bottom row, we're looking at just the oxygen-18 enrichment from the cholesterol. So what we see here is that as we go further into the cell, we see changing domains of enrichment of the oxygen-18. And so the question was, what types of structures are these? Are these related to the endoplasmic reticulum, or are they some sort of organelle that's involved in cholesterol synthesis or cholesterol transport within the cell? But the bottom line is that she had no idea what type of cellular structure she was looking at. And along that same time frame, we've been, and by we, I mean collaborators in Professor Ron Herron's group in Maastricht and Lizanne Oi's group at the University of Wollongong. We were looking at cultured neural networks, and we encountered the same problem where we were looking at 3-dimensional information, and we noticed that lipid distributions were changing location within the cell as a function of depth, but we had really no good idea to determine what types of structures we were looking at. So we need some method to identify cellular structures. So Mary had this idea based on some work that she had been doing with Tamkin's group at Colorado State. And in Michael Tamkin's group, they were looking at yeast cells that were stained with an ER tracker. And they're doing fluorescence microscopy. And they were able to note that this ER tracker, if you look at some of the microscopy images shown in the upper right here, for instance, that the ER tracker, as it was intended to, is specifically located within the endoplasmic reticulum And in this particular case, it's found quite close to the cellular or to the plasma membrane. So Mary's idea was to say, well, what if we use TOF-SIMS imaging using these same types of labeling compounds, and then we have a tag to identify the organelle that we're in, and then we can use that information to do further molecular characterization. So essentially the idea is to repurpose the ER tracker stain, and so the cells that we were studying in this presentation that I'll be showing data for are human embryonic kidney cells that were transfected to express a higher density of Kv2.1 ion channels, and the endoplasmic reticulum, which is typically found bundled quite close to the nucleus. In these transfected cells, the ER lumen is expressed much more closely to the plasma membrane at a higher density and interacting with those Kv2.1 ion channels. And so we have, in this study, the HEK cells that have been transfected and then subsequently stained with the ER tracker, which we're using as a tag for our analysis. So in Michael's group, they did total internal reflectance microscopy, and that's shown in the top panel of images. But for the purposes of the TOF-SIMS imaging, what we're more interested is the actual cell surface, not the one against the glass surface, but the top surface. And so the confocal microscopy images were much more interesting to look at the cells from our standpoint. And those are shown in the bottom row. So in green, we have the fluorescence images for the Kv2.1 channels. In red is the fluorescence image from the ER tracker stain. And then on the right column is the overlay of those red and green images. And the yellow arrows indicate coincidence of the Kv2.1 channels and the ER tubules extending towards the plasma membrane. And the ER tubules are measured here to be between 300 nanometers and 2 microns in diameter, which is well within the range of resolution for TOF-SIMS imaging. So we had some of these cultured HEK cells on silicon substrates, and these, of course, are stained with the ER tracker. On the left top is a total ion image in the positive ion polarity, and the bottom is an overlay of the silicon signal and the phosphocholine head group at a mass-to-charge ratio of 184, showing where the cell is on the silicon substrate. So we zoomed in a little bit and switched to the negative ion polarity to look at this sample. We were looking specifically for the molecular ion of the ER tracker, and in the positive ion polarity, we didn't see the molecular ion. So we switched to negative ion polarity. Again, we did not see the molecular ion, but what we noticed is a high abundance of fluorine signal that was highly localized. And we also noticed a fragment, this is MS1 data here, but we noticed a fragment at a mass-to-charge ratio of 167, and we really had no good idea of what that fragment might be. So we turned to look at the tandem mass spec image, or the mass spectrometry data of that peak at a mass-to-charge ratio of 167. So the full mass spectrum is shown in the top panel, and a zoom in of this mass range down here is shown in the bottom panel. And what we were able to do from the Tandem MS data is identify 3 different components that are present. I should mention that our precursor selection window is monoisotopic, it's 1 Dalton wide, and our collisional activation is at 1.55 keV, kiloelectron volts. So the data here, while the signal is low, this is from an 8-minute analysis collecting a mass spectrum at 256 by 256 pixels, but the signal to background is quite high here, and what we observe is some fluorine signal with quite a low intensity. This is from the total area of that cell. Now, if we zoom in and pull out region of interest spectra, what we notice in the hot spots of the fluorine signal is that the relative abundance of the fluorine signal is increased in these hot spot areas. So potentially, this fluorine signal might arise from the penafluorophenyl anion of the ER tracker. So at this point, we turn to our reference sample, which we didn't do before analyzing the cells because we were quite excited to get going on the cells themselves. So at this point, we turn to a reference sample of the ER tracker. This is the negative ion polarity MS1 spectrum in the top. We do not see the molecular ion, as I mentioned previously, but we see quite a large fragment ion at a mass to charge ratio of 167. And the product ion spectrum of that confirms that it is the pentafluorophenyl anion from the ER tracker molecule. So then the next question is, okay, do we see these same features in other cells that are transfected and stained? We looked at a number of cells, and indeed we do see the same types of hotspots or localization of fluorine signal together with the M/Z 167 peak. And then, of course, we wanted to look at control cells to make sure that in cells that are not transfected and not stained, we're not seeing similar features. Those are all blank. And so we have high confidence that the fluorine and the pentafluorophenyl anion are arising strictly or solely from the ER tracker stain molecules. So if we look at, again, a product ion spectrum from the control cells, we would expect to not see any fluorine in that product ion spectrum for the same precursor mass, and indeed, we do not see any fluorine signal in our control cells. So then going back to the positive ion polarity, we notice that in these same areas where we have localization of fluorine signal and M/Z 167, the pentafluorophenyl anion, in these same areas, we see hotspots of what we believe to be C5H5 fragment, and in TOF-SIMS, we do have enough mass resolution and mass accuracy at low mass to arrive at a compositional assignment. But the question is where is this strange fragment coming from that would appear in these ER tubules? So we go back to our reference sample. In the positive ion polarity here on the reference, we do see the molecular ion of the ER tracker at 580, the sodium adduct at 603. And what we see in the MS1 spectrum is a fairly prominent fragment ion and a mass to charge ratio of 263. Now if we look at the product ion spectrum of the ER tracker molecular ion, so that is shown in the bottom panel here, that's MS2 data set, we see a neutral loss of 318, giving rise to a product ion at a mass to charge ratio of 62. So we believe this peak at 263 is the protonated ion of that 262 product ion. And so if we do Tandem MS analysis on the fragment at 263, this is mislabeled here, this is actually the M+H. What we see in the product ion spectrum is that we can confirm that product ion or that precursor is the dipoxyl dye, the protonated dipoxyl dye arising from the ER tracker molecule. And what's interesting to note here is that while it's small, it's a relatively prominent product ion and a mass to charge ratio of 65, and we believe that this is what we're seeing in the hotspots in the positive ion polarity. So now to the three-dimensional analysis, and I didn't mention earlier, but all of the TOF-SIMS tandem MS analysis that was performed on these cells was done on a 5 nanoTOF 2 instrument. For the three-dimensional analysis, we used a primary ion beam of bismuth 3 single plus, operated in the unbunched mode, which is unit mass resolution. We measured a beam diameter of 80 nanometers, but our pixel dimension for our imaging analysis is 137 nanometers per pixel. And to collect the three-dimensional information, we did ion sputtering with argon 2500 to a total ion dose of approximately 1 e to the 15 ions per square centimeter. So for the three-dimensional analysis, I have a video clip on the right. And so what we can see here is that each of those ER tubules, some of them come all the way in and make contact with the plasma membrane, forming ERPM junctions. Some of them start below or beyond the plasma membrane, so we can see the beginnings of other ER tubules below the plasma membrane. And what's really quite interesting here is that we see within the plasma membrane, if you look at the image on the right, I've made a still, and you can see what appears to be the ER tracker molecule within the plasma membrane. And our hypothesis is that where we have ERPM junctions, it is possible for the ER tracker to be dispersed within the plasma membrane, and we believe that that's why we're seeing a little bit of ER tracker signal within the plasma membrane itself. So summary and outlook. Summarize, of course, if we want to get chemical information, whether it's related to a drug or a lipid, and we want that to be localized to a specific organelle, we have to be able to label the organelle. And so we have a relatively elegant way to do that. There are a number of stains that can be used that have already been developed for different types of organelles, and we believe that those can be applied to look at many different subcellular compartments by TOF-SIMS tandem MS imaging. So in our case, we employed an ER tracker together with tandem MS imaging to directly observe ER tubules and ERPM junctions within HEK cells. That was done at high resolving power, approximately 100 nanometers. Simultaneous collection of both MS1 and MS2 data allows us to do really high fidelity imaging as well as molecular identification, and the same method can be applied to a number of other structures as well. So the next steps that we're looking at is imaging and characterizing the endoplasmic reticulum through the entire cell volume. And along with that, our aim is to see if we can characterize lipids or other types of molecules that might have a higher or a lower abundance inside or outside of different organelles. And by extension, of course, it would be easy to look at the location of drugs or drug metabolites within cells. And thank you for your attention.
Thank you, Greg. That's a very interesting talk. Are there any questions?
Hi, Greg. Nice images.
Good morning, John.
Morning. You had 40 nanometers on your depth scale. How did you calculate that?
So that was, there's a little bit of hand waving involved in that because we did, with the same beam conditions that we use for sputtering, we looked at some polymers, and the sputter rates are similar but not identical, and so it's really an approximation. We did try to do on other cells to measure by AFM the sputter crater after the fact, but the truth is that the roughness of the cells themselves really precluded any meaningful measurement of our sputtered depth.
One other one. I might have just missed it. How were the cells prepared? Were they freeze-dried or frozen?
Yeah, those are freeze-dried, and I don't know all of the details of the sample prep, but it's in the paper that just came out in February.
Thanks.
Well, I think with that, we'll end this session. Thanks, Greg, and to all the other speakers, and for a great session and a great audience.